All. This is Dr. Mubin Sayed from Drbeen.com. Welcome to one more show. So the discussion today is a new study that came out of Oxford, Cambridge, and the study shows how the messenger RNA vaccines have modified synthetic ribonucleotides, how they slip in the ribosome about 10% of the times, and end up uh, causing production of incorrect or mistranslated proteins. So that is a basic study. It is a very interesting mechanism. We know about the ribosomal frame shifts in biology. However, researchers have actually pinpointed when this happens and how does it happen. They have actually looked at the energetics of ribosomes to tell how does this happen. And then they have proposed a fix as well. Like a good biology engineer, they have not only found the problem, but they're also saying, here is the way to fix that. So let's look at this study. So first of all, references. So this is Drbeen.com. If you are interested in getting access to another 700-800 videos, this is your chance. This is your holiday gift. So in the description of this video, there is a link that gives you access to Dr. Bean content, all of it, for only $67. Don't tell anyone, because we have actually, our standard prices are different from this. Okay, so then this is the University of Cambridge, their article about this particular study. Here is the study. N1 methyl pseudouridylation of mRNA causes +1 ribosomal frame shifting, and that's what we're going to talk about today. Then there are some supporting links as well that we might use during our discussion, plus you can use them as you are reading about this study. I cannot thoroughly explain the whole study. It would take hours. However, I would go through enough that then you can look at the paper as well and kind of have your find your way around this. So with this, let's start with my diagrams. So first of all, researchers. It's actually interesting to look at the researchers themselves. So Cambridge University, Cambridge, 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 Cambridge. Then if you see here, they're going to be from Oxford University. Then this is the School of Bioscience, Division of Natural Science, University of Kent, Canterbury. Then this is the NIHR Health Protection Research Unit for Emerging and Zoonotic Infections, Institute of Infection, Veterinary and Ecological Science, University of Liverpool. And then again, Oxford. I believe James is the lead author and Anne is the co-author. So with this, pretty good study, actually. I loved it. So here, I'm going to read some parts of the article and then going to go over the mechanisms of the study. The latest development led by biochemist professor, I'm taking a part of the discussion of the article, Anne Willis, and immunologist Dr. James Theventhiran from the MRC Toxicology Unit at the University of Cambridge build upon previous advances to ensure the prevention of any safety issues linked with the future mRNA-based therapeutics. So they talk about, before this paragraph, they talk about the original inventors of the mRNA vaccine, and then they talk about there are some fixes that need to be done, and they're saying that here are our researchers that are adding those design fixes. And the reason for doing that is, number one, they wanted to understand if the frame shifting is occurring and if incorrect proteins are being formed. And number two, they wanted to understand if these are being formed, then how do we fix it? So that's why they say that for future mRNA-based therapeutics. The researchers identified that bases with a chemical modification called N1 methyl pseudouridine, which are currently contained in mRNA therapies, are responsible for the slips along the mRNA sequence. So you may have heard that mRNA vaccines have nucleotides, or the messages written on them, on that mRNA, are synthetic or methylated to reduce the immunogenicity, plus to increase the durability of these products. So that is what they're talking about, N1 methyl pseudouridine. There are actually other nucleotide bases as well that are methylated. So here is a quote which I really like. This is Anne Willis and her quote: "As billions of pounds flow into the next set of mRNA treatments, it is essential that these therapeutics are designed to be free from unintended side effects." So this is very important. Unintended side effects. Their point of view is that these mistranslated proteins, about 10% of the time, or in 1/3 of the people who receive the vaccine, that is an unintended consequence. And there is a fix. So we should adapt that fix in the designs. That's their point of view. So Nature, we just looked at it. Look at the published date, 6 December 2023. So here is a summary of this study. The summary is in vitro transcribed mRNA. So these mRNA vaccines are called in vitro transcribed, meaning transcription is a process that happens within the cell where from the DNA we make a copy, which is messenger RNA. However, in the case of vaccines, we are not actually taking it from a human being's cell. Instead, we are developing them in the lab. That is why in vitro transcribed mRNA is used in vaccines. So let's say here is a cell. This mRNA is transfected in the cell, right? It goes into the cell, and we've been talking about it from ever. We have lipid nanoparticles, and those my apologies, something happened to my monitor. So we have lipid nanoparticles that are loaded with this messenger RNA. Then those lipid nanoparticles enter the cell. They bring the messenger RNA inside the cell. Once inside the cell, then ribosomes, that are machineries. Ribosome is going to be the main character of our today's discussion. So just keep an eye on ribosome. Ribosomes are the machineries that take messenger RNA and they convert, or they read the code on it, and then they produce proteins according to the code. For example, in these cases of the COVID vaccines, the messenger RNA would contain the code to help produce spike protein. So this ribosome will produce a long string of a protein, which we say polypeptide. That protein will then become folded in the form of a spike protein. However, in 10% of the production, these might get misfolded. And in 1/3 of the human beings, there will be misfolded proteins present. So here, the intended proteins will be produced, plus in 10% of the cases, the ribosomal slips will occur, and we'll discuss that later on, and incorrect or mistranslated proteins will be produced. So that is a summary. And then the researcher said that we have a fix for that as well. The basic fix is the following. They are saying that whenever you have this methylated uridine present in a run, for example, multiple molecules of that are present on the messenger RNA, that is when the ribosome starts slipping. So they're saying that do not have such long runs of methylated uridine, and you can insert some non-cognate messages. You can break them up. That's what they're saying. Okay, so what they did was, once they saw that there is a frame shift happening, and I'll explain that as well, or there's a problem happening, then they verified it as well. So what they did was, this is such a interesting thing. What they did was they infected some mice, or they vaccinated some mice. They also took some humans who had taken this vaccine, and they actually looked at the immune system of the vaccinated mice and human beings and saw if they are making antibodies against these mistranslated, incorrect, garbage proteins as well. And they found that 1/3 of the people that they tested were making antibodies against these garbage proteins too, which was an evidence that these garbage proteins were produced in them. So if they were not produced, then they will not make antibodies against them. And so they verified that in mice and in humans as well. 20 humans, and then 1/3 of them had antibodies against these incorrect or mistranslated proteins. And remember, this is not just one mistranslation. What happens is that imagine if this was the actual protein to be produced, and there is a ribosomal misfunction or slip, then it is possible that another garbage protein that will be produced or mistranslated, as we call it, that might be this much. Then another might be this, another might be here. So various sizes of the proteins might be produced. And those proteins, most of the time, are not going to be useful, but they still have some shape of the original, some part of the original protein as well. Okay, so they found this. I wanted to read out the abstract to you as well before we go into the mechanisms. So if you wanted to just know the summary, then after this abstract, you are good. The summary is done. So in vitro transcribed mRNAs are modalities that can combat human disease, exemplified by their use as a vaccine for severe acute respiratory syndrome. So I mentioned this before, they are transfected into the cell, then the cell does translation and makes those proteins. Then they're saying modified ribonucleotides are commonly incorporated into therapeutic mRNAs described there to decrease their innate immunogenicity. So the nucleotides, we'll see that later in the detail. Nucleotides are small pieces of information that are sitting on the messenger RNA. They say that modifications to them are done to reduce their immunogenicity, to cause less botheration for our immune system to react to it. And number two, to increase the durability of the messenger RNA. But their effects on messenger RNA translation fidelity have never been fully explored. So they're saying that it has never been explored that those modified pieces of nucleotides or messages, when they go through the ribosome, what happens? Does ribosome easily work with them, or ribosome becomes stalled, or does missings or slips? And that's what they found, that there is a +1 frame shift slip. Here we demonstrated that incorporation of N1 methyl pseudouridine into messenger RNA results in +1 ribosomal frame shifting. This is the crux of this whole discussion, that there is a +1. +1 means one nucleotide is missed and the next one is picked up. So to give you an example, if you ask me that, hey, give me this text, and I just miss one letter, let's say I miss the letter T here, and then I combine the remaining text and I give it to you, that text might not make any more sense to you because there is a letter that is missing. So the +1 ribosome frame shifting observed is probably a consequence of methyl pseudouridine induced ribosome stalling during the mRNA translation. So ribosome, and they actually did it. What they did was, as part of their test, this is why I'm amazed at this study, what they did was they had some normal or natural RNA, and then they had the synthetic RNA, and they gave them to various ribosomes, and then they measured the proteins produced by these ribosomes for half an hour. And they saw that the ribosomes that were working on natural messages, they were quickly producing proteins, and the ones that were working with these modified or synthetic pieces, they were stalled and slow, and they were producing less proteins, and they were also producing some broken proteins too. So the +1 ribosome frame shifting observed is probably a consequence of N-methyluridine-induced ribosome stalling during ivermectin, during IVT mRNA translation, with frame shifting occurring at ribosome slippery sequences. However, we demonstrate that synonymous targeting of such slippery sequences provides an effective strategy to reduce the production of frame shift products. So they're saying that we actually have found a solution as well. And that solution is to break down those runs of the methylated pseudouridines. And they're saying that for the future products, this technique should become part of the mRNA design, so the mRNA doesn't produce these incorrect, mistranslated garbage proteins. And of course, and the immune system responds to them. Overall, these data increase our understanding of how modified ribonucleotide affect the fidelity of mRNA translation. And although there are no adverse outcomes reported from mistranslation of mRNA-based SARS-CoV-2 vaccine in humans, these data highlight potential off-target effects for future mRNA-based therapeutics and demonstrate the requirement for sequence optimization. Okay, so summary is done. If you just wanted to hear what they found, we have done that. Now I'm going to go in a little more detail of what exactly they looked at, and not all pieces of it. For example, I'm not going to talk about the exact ribosomal changes that they observed or the time delays that they saw, but I want to go over the main concepts. So here are the details. In our body, our proteins are made from 20 amino acids. There are many, many more amino acids, but our body has decided to select 20 amino acids. Imagine an amino acid being a letter, and we have 20 letters and we combine them in different ways to make proteins. So here I have a couple of letters. For example, this is a structure for a protein glycine, and this is another structure for the protein alanine. And what happens is, when we eat our food, we get these amino acids. Those amino acids are present in the cytoplasm of the cell. So they're stored in the cytoplasm. They're just present in the cell. So then the cell gets the messenger RNA as a recipe, and the ribosome is the machine that reads that recipe and makes the protein. So what that ribosome does is it picks up these individual bricks, if you will, individual letters, if you will, combines them as a polypeptide and makes a long chain of a protein. So here, this would happen in a ribosome. So glycine plus alanine are put together by ribosome by a peptide bond, and water is removed from these two molecules. So here is glycine, alanine, and now we have glycylalanine. But this is just two amino acids. Imagine thousands of them put together in a chain, then folded together to form a protein. Now the messenger RNA are the carrier of the information to make these proteins. So what happens is that messenger RNA has nucleotides on it. Nucleotides have this little ribose here, and then some phosphate plus a base. Base can be adenine or guanine or cytosine or uracil in case of RNA, or thymine in case of DNA. So this is basic biology that we've all done. So these are the bases. So imagine that for every amino acid, there are 3 nucleotides that will come together and provide the amino acid that represent an amino acid. So I'll give you an example. I have a table here with those amino acids. So here, for example, if uracil and uracil and uracil, 3 of them, they make a codon, 3 messages together, nucleotides together, they make one codon, and one codon codes for one amino acid. So for example, UUU is making phenylalanine, UUA is making leucine, and as you can see, there are various amino acids and their codes for nucleotides. So back here, these are various letters, and if you see here, this is the messenger RNA. Messenger RNA has a beginning and it has an end, then it has some untranslated regions, and then it has a region which can be translated into a protein, or that there is a recipe in the center that can be read to make a protein. So here, for example, these three bases or nucleotides, U, and U, together will code for phenylalanine. UUC also codes for phenylalanine. Then CUU codes for leucine, GUA code for valine. So if you can imagine where it says GUA, this molecule with guanine is sitting here. Then this molecule with uracil is sitting here. Then this molecule with adenine is sitting here. That is how there are tiny structures that are stuck on the messenger RNA. Now the question is that, all right, we have these codons present. How do we read them and make proteins? So what we do is we have ribosomes. I'm giving a very high level biology class. So we have ribosomes. We stick this messenger RNA and we attach it to the ribosome. Then what happens is ribosome, this is important to understand the frame shifting. Ribosome read three nucleotides at one time, or one codon at one time. So UUU will be the first one that will enter the ribosome. Ribosome will then have these trans tRNAs. There are another set of enzymes that are running around, and as the ribosome needs a specific amino acid, these guys are going to run, capture that amino acid, and come and attach here and provide that amino acid to the ribosome. So, for example, phenylalanine. Once that is produced, then the ribosome will stitch it to the next one, and then it would keep shifting the RNA, keep receiving a new amino acid, keep stitching that amino acid to the old So here is what happens. Ribosome, instead of these three, it has already put them together. Now it has moved to the next one, then it has moved to the next one. And then what happens is, as the ribosome traverses three nucleotides at a time, or a codon at a time, it continues to make peptide chain. And that chain, do you know that there is a tiny little protein that is sitting next to the ribosome? And as this chain is being formed, this polypeptide chain is being formed, it actually starts accumulating in this protein. This is called a heat shock protein. It acts as a chaperone, and the protein, this little thread that is being formed, starts accumulating in this protein, and that protein then helps form and fold this newly developed chain into a three-dimensional structure. This is happening all the time in our cells. So once that is done, we have a protein. Now, here is an example of +1 frame shifting. So it is upstream frame shift, or basically, let's just say it in a simple term, out of these three, imagine ribosome could not read one. And what does it do? It just reads two, and then it could not read this one. Let's say it just read the fourth one. And now, because it works with three at one time, it read UU, and the fourth one is U as well. So luckily, in this case, it would still make phenylalanine, but then, or attach, but then it would read U, C, and C, and that would be an altogether different amino acid. And all of a sudden, we have a mistranslated protein. So here, I've given an example that imagine here we have AUU, which is for isoleucine, and then we have GGU, which is a codon for glycine. And imagine that when ribosome is reading, this U here is methylated uridine, synthetic uridine. And this, in 10% of the cases, imagine that these are all synthetic uridines, the U's. In 10 out of 10, one of them is going to slip. So imagine this one got slipped. So when this got slipped, now ribosome is not reading AUU. Instead of that, it is reading A and U and G, because this guy slipped. So now instead of making isoleucine, the ribosome is now looking at A, U, and G, and AUG is methionine. So all of a sudden, we have a different amino acid inserted in here, which was not supposed to be. Many times, it's just an innocent thing, and one amino acid change doesn't do a lot of damage. But a lot of time, that damage occurs, and sometimes the protein translation stops here. Sometimes it stops a little later. So we would end up with the garbage protein. So now imagine that this was the vaccine mRNA, which is slipping in the ribosome, and now it is ending up making incorrect proteins. Now, those incorrect proteins are, of course, also proteins produced by the cell, and the cell would present it to the immune system, and the immune system would start making, the reaction to those garbage proteins as well. So, after this mechanism, I wanted to read to you some of the comments from the authors in their manuscript. So they said, this is amazing. They said, we then compared interferon gamma response to predict +1 frame shifting for SARS-CoV-2 protein products in 21 individuals vaccinated with Pfizer-BioNTech, and compared those responses to those of 20 individuals vaccinated with ChAdOx1, which is the vector-based vaccine. Why did they compare it to this one? Because the vector-based vaccine, or the adenovirus-based vaccines, do not have synthetic mRNA. So their mRNA is natural, and because of that, the protein they form is different, or the actual spike protein, compared to Pfizer-BioNTech, or I would say Moderna as well, where the proteins are malformed as well because of slippage. So what they did was they looked at interferon gamma production of these human beings against the trash products. And they saw that 1/3 of the Pfizer recipients were responding or had antibodies to mistranslated proteins. We detected a significantly higher interferon gamma response to +1 frame shifted antigen in Pfizer-BioNTech vaccine group compared to the ChAdOx1. There was no association between T cell response to +1 frame shifted antigen and age, sex, or HLA subtype. So meaning it was not something else that was doing it, it was the methylation that was causing it, and it was causing it uniformly regardless of the age, sex, etc. Both vector-based vaccine and Pfizer-BioNTech vaccine produced response in frame of SARS-CoV-2 spike. So there were correct spikes formed as well. So there was a response for the correct spike, but response to +1 frame shifted products were observed only in individuals with vaccinated by Pfizer-BioNTech. Then they said, how did this happen? Why would that +1 nucleotide would get slipped away? So they said, it seems like that the ribosome, the machinery that is using reading and making it, it has hard time working with the synthetic or methylated nucleotides. And its energetics change. They've given a complete explanation of that. I'm not presenting that here. But the end result is that the energetics change of the ribosome, it then slips on it. Not only that, it actually takes more time to work with it. So the time durations also change. That means the stalling of the ribosome occurs, and then finally ribosome slips. So together, these data show that slow translation of 1-methyl pseudouridine mRNA is possible due to ribosome stalling, which is caused by altered amino acyl tRNA binding, and which can be rescued by increasing the incorporation of near or non-cognate amino acid into elongation peptide. So what they're saying is that if this was the mRNA and these were methylated, nucleotides, if we insert non-cognate amino acids, that means amino acids that don't exist. So we just insert little breaks in them that actually do not translate into anything. This is like empty spaces. If we do that, then the ribosome doesn't have hard time working with these methylated nucleotides. But if you just have a huge run of methylated nucleotides and you ask the ribosome to go through all of them, the ribosome starts slipping. So that is the solution. Create empty spaces between them, among them. And they're saying that we expect that in the future products, this should be incorporated as a design fix. So they talk about it, that why is it important? Although there is no evidence that frame shifted products in human generated from Pfizer BioNTech vaccination are associated with adverse outcomes. For again, this is their statement. I don't think they looked for it. I don't think there are any studies that looked for these products. Maybe now people would start doing the studies. So we can say that at this time, there is no knowledge of these products causing harm. We know that the vaccines can cause side effects, but how many of those side effects are because of these? They are saying we don't know. For future use of mRNA technology, it is important that mRNA sequence design is modified to reduce ribosome frame shifting events, as this may limit its future use for applications that require higher doses or more frequent dosing, such as the in vivo production of hormones. So they're saying that if mRNA technology is going to be used multiple times more frequently in higher doses, then this fix should be made. Otherwise, the people would end up getting lots of trash, wrong proteins. And not only this, that there are proteins that are wrong. Of course, our immune system, when it would see a bad protein, it would start attacking it as well, just like it would see a good foreign protein like spike, it would attack that too. So now our immune system, how would that react? What will happen? They actually talk about it here. It is important to continue investigating therapeutics, mRNA translation, and immunogenicity, as the evolution of antibody and cytolytic T cell response against plus one frame shifted spike variants and peptides has not been systematically evaluated. So they're saying we should continue trying to research this area, because we do not know what will cytotoxic T cell do. Will they learn a bad, trashy protein, mistranslated protein, and go and attack some part of our own cells because of molecular mimicry? Will these bad products that were not intended, that we did not even foresee when they would occur, will they train our immune system to start attacking some part of our own body? So that is what they're saying, is that it is important to make sure that this doesn't happen, because we do not know what cells will do with that. So that's why they're saying T cell response against plus one frame shift spike variants and peptides has not been systematically evaluated in humans, and ELISpot responses obtained from the pooled peptide may also underestimate T cell responses. The main in-frame mRNA encoded product is unlikely to elicit an adaptive immune response, but this is very important. So this statement has to be clarified by understanding why methylated nucleotides were used. The methylated or synthetic nucleotides were used not only for the durability, but to reduce immunogenicity. And what immunogenicity? Against the innate arm. The vaccine designers did not want the innate arm to quickly attack the vaccines. So they changed the mRNA nucleotides. What they're saying is that fine, the protein that is produced that is whole, and this messenger RNA may be prevented from being attacked from the innate arm. But what would happen when the cell is producing these abnormal proteins? Will the innate arm still be sitting on a side and say, okay, you know what, I'm not going to touch this? So the main in-frame mRNA encoded product is unlikely to elicit an adaptive immune response. Sorry, did I say innate? Adaptive, that is B cell and T cell. Sorry, scratch my last statements. But presentation of plus one frame shifted products could activate T cells and target host cells. Target host cells. We reasoned that if we were able to identify plus one ribosome frame shift sites or sequences, it would be possible to later to alter the mRNA sequence to reduce such effect. So then they continue and they say how they changed it. So how did they fix it? This is from the article from the Cambridge University. When the ribosome is confronted with a string of these modified bases called N1-methyl pseudouridine in the messenger RNA, it slips around 10% of the time, causing the mRNA to be misread and unintended proteins to be produced, enough to trigger an immune response. Removing these runs of N1-methyl pseudouridine from the messenger RNA prevents off-target protein production. So the two important keywords here, runs of the methylated products and off-target protein production. So this is the discussion. Thank you very much for your time listening. I hope that it makes sense. Please like, subscribe, and share. I'm looking at the comments here as well, so that's why I go here. And in the description of this video, if this is the kind of work that you like, explanation of the mechanisms, regardless of what the product is, then please support me. And you can support, the best support is to buy drbeen.com. There is a link in the description, and get subscription there, or get one-time entry fee and become a member there. We have CMEs available there, CEUs available there. We do weekly new videos there. In addition to the videos that are on YouTube, there are separate premium videos that go to drbeen.com as well. Otherwise, there are other links as well if you would like to support this. And thank you very much. Have a nice weekend, and I'll talk to you later. Like, subscribe, and share.